Thank you, Deku from Auckland University, and today I will be sharing with you part of my work during my PhD, focusing on the fungal communities associated with metaverse infection and the use of microparasites and potential biocontrol agents in human disease. So I think we can start my presentation with these images, but that's what I witnessed firsthand during my sample collections. The native trees are like dying because of weather rust, and it's deeply check my motivation to find a microparasites or something like environmentally friendly solution to have these plants in the battle against the disease. So for the sample collections, the lead samples were collected on two host plants. The first one is Metrocytos aerosan, or one, but you can see just like outside of the windows. So this plays a very important role in the cultures as well as the ecosystem, providing the nectar source for the native birds and also serves as a coastal erosion buffet. And the second host plant is or Ramamara. And this is a key component in the native forest, providing the habitat and the food for the native birds and pollinators. And also the essential is also contain the potential antimicrobial properties, and unfortunately, they both host plants are considered and highly susceptible to motor rust. And so, in summary, the lead samples were collected from 8 sampling sites across New Zealand in 4 locations, and including some of the native forests in Taranaki and Rotorua, as well as the urban areas in the Christchurch and Auckland. So following the field surveys, the total genetic DNA was extracted from the lead samples, and then only 5 of the ITS regions were in the first PCR, and then the first PCR products were indexed with specific barcoded primers in the second PCR, and then the applicants were sequenced using the MySet platform, and the data was analyzed using the bioinformatics tools to see how the fungal communities look like. So here's some of my results for the alpha diversity, which measures the within sample diversity. There was no significant difference between host plants and the environment. However, by looking at the beta diversity, which measures the difference between the groups, the results show that there's an overlaps, which indicates while most of the fungi were shared between the groups, there were also some fungi unique to the specific host plant or the environmental setting. So when looking at the fungal community composition, there was a noticeable difference between host plants. So in Pohutukawa community, fungal community, there's some of the fungi such as the Crafoliola, Germania, and Pseudozyme at higher relative abundance. In contrast, in the Rama, Collectotrichum, Elecnogia, and Libertas meses were the dominants. So the differences are also observed between the different environments. So as you can see, in the forest, the environment, Collectotrichum and the Pseudozyme costomer, which is shown in the red color, and the dark green, had a very high relative abundance. So in terms of the drug pathogen or Puccinia, which is shown in the gray color, there was a higher, not visible higher relative abundance in Pohutukawa compared to Ramaraman and in the urban area compared to the forests. So as I mentioned earlier, the most important goal in my research is to find microparasites and isobial control agent against the rest. So this is involved in the fungal isolation, identification, and also the numbers of bioassays. So in total, the 208 fungal isolates were recovered, and we only 64 genera, and there was a higher population of fungal isolates and recovered from native areas compared to urban areas, and the most frequently recovered fungi were like collected dragon, fusarium, Beta-Sphaeropsis, and Bilitum. So based on the research on the little region, 23 cutative microbial were selected, and they have been reported as biocontrols of the rust or other plant diseases, and they belong to 11 genera shown the list on the left, and they are culture morphology is showing the pictures on the right. So these 23 isolates were initially screened for their antagonistic activities using the intro detached branch assay. So in this assay, the branches were firstly inoculated with the rust and then putated microbial, and 14 days after the inoculation, the rust culture was like examined under the microscope, and the extent of colonizations were calculated. So here's some of the results showing the wide range of variability from like 4 to 35%, and serum of them, which is significantly different from the culture, was selected for the further test. So in this germination test, I would like to test if this isolate can inhibit the germination rate of the rust spores. So in this assay, the rust spore suspension was mixed with like spore suspension of test isolate, and then incubate at the right condition to facilitate the germination of the rust spores, and then examine under the microscope observations, and 3 of them which had the highest ability was selected. So for the further consideration as a biocontrol, I would like to test if the isolate took out any harm to the plants, and 2 of them, which is in 3rd and 4th one, were like considered as non-pathogenic, developed with no symptoms developed on the news, but unfortunately one isolate caused some of the lesions on the legs, and this isolate was excluded from the further consideration. So in summary, from the capsule collection of 208 isolates, 2 isolates successfully passed through all the bio assays, and they were molecular identified that Trichoderma hasianum and Cladosporium elucidated by sequencing of not only ITS region, but also other taxonomic genes like TEF1 or LSG. So the isolates were then tested under the implantar conditions, and the experiment was designed in randomized controlled approach with 9 active patients, and using 2 application methods, which is preventive application, which applied the putative microparasite 2 days before the rust, and curative 7 days after the rust. This is a picture of myself working in a PC2 glass house. So it's rust Myrtle body is still considered as a new and registered body in New Zealand. So all of the work has been done under highly contained facilities, electricity, electricity greenhouse, and people working with Myrtle are still required to wear the full protective with gear to prevent any unintentional spread of the disease. So the implant experiment showed that the preventive treatment significantly reduced the disease severity index by around 19% compared to 30% in the curative applications 3 weeks after treatment. And in addition, the plants treated with trichoderma showed the promotion in the blood health with more new and healthy growth compared to the plants treated with Sphaerosporum or the control. And I also conducted some of the work with a scanning electron microscope to investigate any interactions between putative microorganisms and the vesicles. And here is exciting part during the PhD, which shows some of the indications of interaction with the coiling of the fungal hyphae around the vesicles, or the information of the vesicle structures upon the contact. So the takeaway message. So for the fungal community's proposition value between Australasian environments with a higher relative abundance of modifiers to improve together and urban area environment. And 23 fungal isolates were identified putative microorganisms, and only 2 of them showed the strongest antagonistic activity in vitro assays. And in the blood conditions, preventive applications significantly reduce the disease severity compared to curative treatment. And for ongoing work, currently analyzing the metabolism data to investigate the changes of the fungal community after the treatment, and also doing some of the DNA quantification to evaluate the persistence of the fungal isolated beliefs using the qPCR. And lastly, I would like to thank the Universe program for the funding, and also thank you, Michael, Kirsty, and Bernard, who are here today for the help with the sample collection, and also thank you, Australasian Plant Pathology Society, for the pursuing so I can attend this conference, and thank Monica for technical help. And special thanks to my supervisors, Vivian, Haley, and Sumi, for their interaction and guidance during my PhD. And thank you for your listening.